Good morning, everyone. My name is Wenwen, and I'm come from Nanjing Normal University. I'm glad to participate in international conference on agriculture, fish, and marine biology. The title of my paper is Molecular Characterization of CIRBP from Takifugu fasciatus, and its potential roles is clod-induced hepatocytosis. My supervisor was Professor Zhang and Associate Professor Zhang. Next, I will report and display the contents of this paper. As percent, environmental change has intensified, and extreme long temperature occurred from time to time. While fish as sustainable, the environmental temperature directly or indirectly regulates the growth, reproduction, and the development of organisms and other processes. Extreme cold winter bring problems such as high overwintering mortality and increased overwinter costs, which seriously restrict the healthy development of farm, fish farming. Moreover, the puffer fish is a typical majority fish and the majority is between land, river, and the ocean, and it is more weak to temperature changes. Several genes have been identified and that are activities during long temperature stress in fish, such as the cold-inducible RNA building protein and the anti-freezing protein and the high mobility group box 1 protein. It is correctly studied in species such as toothfish fish as well as large yellow croaker. Coding invisible binding protein is an important mRNA molecular SHARP1 protein that can be induced to overexpressed by temperature and belong to the family of RNA building proteins, and which can be widely expressed under the stress of environmental factors. And the expression of CIRBP increased significantly under conditions of long temperature lines and change in osmotic pressures. And we in the studies the four less sequence of CRBP CDN are called tolerance genes of published pufferfish, which can clone and bioinformatics analysis was carried out. The expressions pattern of tissues distributed was investigated, and the binding mechanism of CRBP-TRX was verified, and the effect of long temperature stress on the comprehensive physiological and biochemical cas- uh, cascade of the pufferfish, such as apoptosis and immunity, was investigated. Two proofs scientific based for the molecular breed of new long-temperature tolerant pufferfish. We design two vectors experiments of time and temperature using 13 and 19 degrees Celsius as the long temperature stress temperatures, and the sample was collected at 0, 6, 24, and 1960 hours, and then equations analysis and functional analysis was investigated using liver tissues of pufferfish as the study object. We clone the CRBP genes and analyze the biological information and found that the fullness of the gene is 424 BP and an open reading frame of 16 hours 120 4BP, coding for the uh, sequence of 174 amino acids. The protein contains are conserved RRM schedule domain. The great part in Figure C is the binding region that was predicted to be able to blend the RNA by online software. Amino acid sequence similarity results showed that pufferfish CIRBP has the largest amino acid sequence homology with the tiger puffer, followed by those with spotted catfish and zebrafish, suggesting that it is relatively evolutionary concerns with the conclusion was also supposed by the physiological tree result. The result of convenience analysis showed that the upstream and downstream genes of CRBP genes of the pufferfish and the tiger puffer was very convenient. There was also some similarity of humans and mouse. The expressions of CRBP genes in different tissues of pufferfish was analyzed by semi-quantitative and real-time fluorescence quantification. The results showed that liver tissues has the highest expression and muscle has the longest expressions. The liver is the most important organ in energy metabolism. Therefore, we chose the liver of pufferfish as the target of the experiment. The live tissues was distillate at 24, 19 and 13 degrees Celsius respectively, and the liver tissues was found to be congested under long temperature stress, and there was many black lesions on the surface at 30 degrees Celsius. Similarity, the histopathological result revealed that with the duration of cold expressions at 30 degrees Celsius, the liver was damaged with varying degrees of vascularization. Meanwhile, at 1960 hours, the liver detected several damage and malfeasance as virgin outlines and sold by the neutral. To evaluate the liver oxidative stress response induced by cold exposure, the level of RS at 13 degrees Celsius and the activities of antioxidant analysis was measured. Compared with the fish exposed under 13 degrees Celsius immediately, the RS fluorescence signal intensity noticeably increased after 6 hours and 24 hours treatment. However, the fluorescence intensity at 16 hours was significantly dropped compared with the 24 hours clothing exposure. Moreover, the activities of antioxidant analysis including SOD and CT, per 10% the upper regulate trends subject to long temperature, and the change was more obvious at 1960 hours. Then the contents of MDA and high GG cider was also stronger augment after more than 24 hours of cold treatment. The equations of apoptosis-related genes and further explored. BCL2 is an anti-apoptotic gene, and BX is for apoptotic genes, and the radio of these two genes is a key factors in determining the degrees of apoptosis, and a disease in the radio of BCL2 to BX indicated increase in the level of apoptosis. We further measures the mRNA levels of inflammation-related genes to evaluate the effect of cold expressions on inflammatory response in pufferfish livers. NLRP3, ASC caspase 1, and IL-19 in mutual genes was all significantly higher than the control group under long temperature stress, suggesting that the more the temperature dropped, the strong and physiological stress it induced, and that the took certainly measures to cope with the stress. Similarity to expose apoptosis, we made tunnel sections of liver tissues of pufferfish under long temperature stress, and expose apoptosis by fluorescence intensity, and we found that the fluorescence intensity of tunnel strands positively increased from 0 to 24 hours of on-temperature treatment, and then decreased insignificant at 1960 hours. It indicates the publish adopt a series of countermeasures to alleviate the stress caused by the long temperature under prolonged long temperature treatment. Given the evidence that increased ROS would activate the TRX-TNF alpha signal and lead to subsequent apoptosis that further aggravates liver damage, and we evaluate the effects of ROS on TRX-TNF alpha activation as a temperature disease. The findings suggest that mRNA levels of CIRBP, TRX, TNXI, ERK, and TNF-alpha were significantly increased at 19 or 13 degrees Celsius with more than 24 hours cooling treatment when compared with. Based on those results above and previously studies, we speculate that CIRBP could binding to packet 3 UTR of mRNAs and regulate these expressions. Then we performed WB and found that the expressions of TRX protein was accumulated in cold expressions showing a similarly profile with the CIRBP protein. Although immunohistochemical streaming showed that both the CIRBP and the TRX levels of the pupfish under cold chain was markedly increased in time and temperature-dependent manner. Together, those results suggest that TRX is a potential downstream target of TRX. Code location section analysis of CIRBP and TRX showed that TRX was predominantly expressions on cytoplasm, while CIRBP was distributed in both the nucleus and cytoplasm. We found that CIRBP may regulate the appearance of TRX through past transportation way. To confirm this assumption, we carried out independent RAIP experiment followed by RT-PCR, and the results indicated that mRNA levels of TRX were significantly upregulated after the additional of CRBP compared to control groups. This change was more noticeable under cold expression. Taken together, our results indicate that CRBP can bind to TRX mRNA and facilitate its appearance at the host transportation levels and consequently alternately hepatic homeostasis. Okay, that's all I have said today. Thank you very much for listening.